Hi, everyone. Can anyone put in the chat if they can hear me and see me? Okay. Okay, great. So welcome. Good afternoon. And thanks for joining this introduction to CellProfiler workshop. I already put in the instructions for the workshop that it's preferable that CellProfiler is already installed. And if it's not, I am going to share the link where the software can be downloaded from in the chat box. And I'll also be putting in the chat where the other material that's required for the workshop can be downloaded from. So I'll just go ahead and do that first. This is the link to download all the workshop material, including the slides that I'll be sharing and the link to download CellProfiler. Great. So with that, I would just get started with today's workshop. Hi, everyone. Again, I'm Shatavisha Dasgupta. I'm a postdoc at Lab at the Broad Institute. And helping me with today's workshop would be Paula Lanos and Suganya Sivagurunathan, who are also postdocs in the same lab. And we also have Beth. I'm sorry, I don't have her name on the slide. Anyway, so for today's workshop, I would be first of all going through some of the basic concepts related to image analysis and also discuss what are the basic components of an image analysis pipeline. And as I go through that, I would also be showing you the CellProfiler software interface, where to find the buttons that will help you create a pipeline for your own projects. And finally, as a last step, we will be going into breakout rooms, where we'll perform some hands-on exercise and actually build a CellProfiler pipeline from scratch. So to start off with a bit of background, as we all might know, image analysis has not always been quantitative. Biologists over past several years have relied on manual annotation, measuring things using reticules in their microscope, using very tedious, time-consuming processes, which are not just time-consuming, but then can also be error-prone and suffer from lack of reproducibility. However, in the recent years, we now have excellent microscopes which produce these images that contain a wealth of information within them. And these digital images are actually composed of pixels, which are nothing but rows and columns of numbers. But to extract the relevant biological information from these numbers, we need robust analytical tools. And why? Because unfortunately, human brains are not built for quantification. So if anyone in the audience is not already familiar with this image, and if I tell them now that this is actually a grayscale image, I don't think anyone will believe me unless I show them this inset, where you can clearly see that this is a grayscale image with some colored lines superimposed. Our brain is actually filling in the rest and turning a grayscale to a color image. More examples to prove the point that our eyes play a trick on us all the time. Nucleus of A is not brighter than nucleus of B, and C and D are actually of the same size. So automated image analysis is not just faster and less tedious. It also helps us measure multiple different properties within our cells of interest at the same time, and also identify very, very subtle changes which our eyes cannot pick up. In recent years, we have a lot of advanced technologies such as single-cell RNA sequencing come in for measuring different biomarkers or characterizing cell types. But one thing should always be kept in mind that a lot of these technologies are dissociative in nature and they lose the important spatial or a localization context. So to make this point clearer, I have these two images up here, where if one is measuring the mRNA or the protein levels, it just seems that these two groups of cells are very similar to each other. But if we look at the images, we can see that here the marker is expressed in cytoplasm and here in the nucleus. And so they are, after all, not so similar to each other at all. Also, as researchers, we want to share our data that's generated from the images by creating these nice visualizations for publication purposes. And so for this also, we need robust analytical tools. So CellProfiler is one such free and open source image analysis software. It's been around for quite some time now. And as this graph will tell you, it's being cited several times over the previous years. And this platform has also been, the software has also been ranked as one of the most flexible and easy to use. And all of this has largely been possible thanks to an amazing team. And here are some of the folks who have been part of this team in the past and some of the current members of the team. So CellProfiler is a great software, but it's of course not the only one that's available. Bioimage analysis is complex and challenging, so obviously we need a lot of tools for that. And other very popular open source tools include ImageJ and Fiji, which are well known for their speed and versatility. But a slight downside for these tools is that they require some familiarity with writing scripts or macros. And yet another amazing tool is Elastic, which is actually quite popular for segmenting hard to segment images, such as bright field images or tissues. But again, this tool is mostly geared towards identifying objects of interest rather than quantifying metrics from these objects of interest. So having given that introduction, let's just now take a look at how the CellProfiler interface looks like. When we open up CellProfiler, this is the welcome screen we get, which is actually quite handy because it contains links to some example data sets which people can use to build up their expertise for creating pipelines. It has links to tutorials. And last but not the least, it also has a link to a Q&A forum, which is forum.images.sc, which is a great place to ask questions, not just related to CellProfiler, but related to any other image analysis software. This is how the CellProfiler general interface looks like. On one side, we have the pipeline panel, and on the other side, we have the settings panel. On top of the setting panel, we have the space where we can record our notes on whatever changes we are making to a pipeline so that we can refer back to those at a later time point. And as I mentioned, CellProfiler is a very user-friendly tool. So there are friends hidden within the software itself. And these are these question mark buttons, which show up everywhere. And if we click on those, it will not just give information on the modules or the parameters that go into a module. They also contain sometimes linked to relevant references, which are very good reading material. So I would start off with what shows up at the top of the pipeline panel. And this is known as these are known as the input modules. These comprises images, metadata, names and types and groups. And what the input module basically does is it creates image set. What is an image set? It's basically an image split into its different channels. So for example, here the DNA and the GFP channel images will be constituting one set. So input modules are quite complex to grasp, and I would recommend for a more detailed understanding to go to this link. And there's actually a great blog post and also a link to a video tutorial, but to give some sort of an idea about what the components of input modules comprise. It contains images, metadata, names and types and groups. And these are basically the parameters with which the software understands what type of image is being provided to it. It extracts the relevant information such as number of plates, which wells are present in that plate, whether the image is 2D or 3D, and whether if there are multiple plates in the image set, whether they belong to certain groups or not. Not every field is mandatory. For example, image is a compulsory one, and also names and types, but metadata and groups, these are considered optional fields. Also important to know at this point is, unlike the CellProfiler pipeline, which progresses sequentially through the steps, these are not linear. So if the parameters that are entered in a subsequent step is not compatible with the previous one, the pipeline will break. And this is basically how the input modules, the settings panel of the input module look like. This contains a section where the images are to be added. It's totally possible to just drag and drop only the images, but even if the folder contains images and some other files, we can mention here how to filter the contents of the folder so that only the images will be taken into consideration for running the pipeline. This is what comprises the metadata module, and here in the drop-down we have the examples that can be the parameters that can be inputted from where the software will derive information such as how many channels there are, what plate, the plate number, the sites and wells. And how this information is extracted is by the use of some regular expression, which will be used to match the file name. And from this, I guess folks might be already guessing that this requires following some sort of naming convention of the files and the folders for the function to execute properly. And this again is provided, the details are provided in the link to the blog post that I just mentioned. So names and types, which is a compulsory module, is where we mention the channel names. We mention whether the image is a 3D or a 2D one. We indicate what type of image it is, for example, grayscale or color. And once we click on the update button, all of these information will populate in the table that's below. And finally, groups, as I said, is not a compulsory parameter, but if there are, for example, multiple groups of plates present in the experiment, this is where we would need to click and then choose whatever parameter will go into the metadata category. So having looked at the input modules, let's take now a look at the modules that go into the actual pipeline that will analyze the images. And this will look like this with all the different modules lined up next to each other. How to add these different modules to the pipeline is, for that we need to go into the adjust modules and click on the plus button, which opens up a window like this. Now, CellProfiler is an amazing tool with many, many different modules, and they can be broadly grouped into file processing, image processing, object processing, measurement, and so on, various different categories. Again, to reiterate, module help will provide detailed information on what each of these modules does. But in general, image processing and object processing are essential components of any pipeline. But again, what module a pipeline will constitute is mainly dependent on the biological question that the researcher is trying to address. And specific modules can be searched for in this search tab using keywords. So once we have added a number of modules to a pipeline, the next step would be to run the pipeline in a test mode. And why do we do that? This is just to check whether the modules have been added in a sequence that is compatible to each other, all the parameters have been inputted well, and also the final output that's being generated by running the pipeline is exactly what we were hoping to, is answering the questions that we were hoping to address by running the pipeline. So once we start the test mode, some additional buttons appear. For example, the run and step. Running will make the whole pipeline run sequentially. And by clicking on step, it will make sure that the pipeline progresses through one step at a time. In addition to that, next to all the modules, some additional icons such as the eye and this pause button and the green play button will appear. The green play button shows which module will run the next, and that module will also get highlighted by an underline. If the eye is turned on, it will show the output after running the module. And if we just click it off, then the display won't be shown. And if we don't want to run the entire pipeline in the test mode and we only want it to run till a certain point, we can also click on this pause button, and then it will execute only the steps that precede the pause button. And Also important to remember that this attention icon will show up every time in the test mode. That's because the results of the test mode will just appear on the screen in a window, but it's not exported to a spreadsheet, unlike when we are actually running the pipeline. And last but not the least, if there's some parameter which is not compatible with the running of the pipeline or the module, this kind of an error button will show up and the pipeline will not run. But the great thing about CellProfiler is if we hover with our mouse over that error button, it usually tells us where exactly it's encountering the problem and how to, and we can figure out how to fix it. So when we open up individual images in the CellProfiler interface, this is the image window that we get. Again, good to know tools that are present here. The magnifying glass is for zooming in and out. The backward arrow will just take us one step back or just zoom out a little bit. Home button will set us back at the first position. And there's also a ruler which can allow us to measure objects. For example, here a nucleus, the diameter of a nucleus is being measured. Also, just hovering over the objects in the image will let us know the intensity, and these measurements will just show up in the bottom panel as shown here. So with that basic information, I would then go on to describe what the steps of a pipeline usually comprise. So our first objective for analyzing any images would be, first of all, to identify our images of interest. And this generally follows the basic premise of my object of interest being the brightest in the image and everywhere else being dark. But as we know, in practical experience, this is not always the case. There can be variation in staining. The signal to noise ratio is not always ideal. There can be background signal as well. So for that, to be able to identify our objects of interest accurately, we need to make the distinction between foreground and background optimal. And so for this, we process images. Important to keep in mind is just for segmentation purposes, any transformation that we can apply for processing is considered legal, but this processed image should only be used for identifying our objects of interest or segmenting. Because every time we are processing an image, we are adding or subtracting intensity values. And if we use the processed image to then measure the intensity in subcellular compartments, we may get spurious results. So it's important that we are measuring the intensity from actually our original images and not the processed images. Once we have performed transformation and segmentation, again, to evaluate how the segmentation is actually performing in the software, we need to include modules such as overlay outlines or overlay objects. And this will give us an idea of how much more fine-tuning needs to go into the segmentation. So again, there are several various options for image processing in CellProfiler. And again, this is not the exhaustive list, but the ones that are used usually most commonly are enhance or suppress features, cropping, or color to gray modules. So how do we find our objects of interest? This is done through a module called Primary Object Identification in CellProfiler. The basic principles behind this would be, first of all, to distinguish the foreground from the background. And also, once an object is identified, we need to figure out whether it is actually one object or two objects which are touching each other, and so to split or merge objects accordingly. So for separating the background from the foreground, the method that's used most commonly is called image thresholding. So for this, the intensity values of the foreground and the background are plotted in these in this kind of a histogram. And as we can see that there are some values at the lower end, and then there are some values at the higher end. And the cutoff that will actually separate the foreground and the background will usually lie somewhere around here. Moving this cutoff to a lower value will mean that we would be including some of the background in our object of interest, and moving it too high will mean that we will be actually losing out some parts of our object of interest. So we need to just find the ideal cutoff that will make sure that we are identifying our objects of interest ideally. There are several different principles and methods of doing this. So the one that's used very commonly is called OTSU, and it's particularly helpful for images where there are actually multiple peaks. And for example, if the object of interest has two different intensity peaks, OTSU is usually the option to go for. For those of us who study the fish dots in the cells or bright speckles, then robust background would be a good option to choose. And this is how, when we open up the Identify Primary Object module in CellProfiler, where we can choose the thresholding method depending upon the type of image that we are trying to analyze. Also, before performing thresholding, sometimes a transformation is applied that's called a log transform, which can be helpful in a lot of different cases, because, for example, where the histogram is not very ideal for choosing a threshold, by performing a log transform, it will help to identify these cutoff values easier and just make thresholding easier in general. So once we have separated the foreground from the background, the next thing to do would be to split or merge objects so that things can be identified separately. For us, when we look at images, for example, here we can see that there are some objects which are touching each other. We can tell that these are separate ones, but how does a software do that? So what it does is it can use two different principles. One is called an intensity-based and the other one is called a shape-based. So intensity-based method usually works well for smoother objects, which tend to have a lot of intensity in their central part and are dimmer at the edges. And so here, the peak values of the intensity would be used to determine where the boundary between the two objects lie. But for objects that are kind of lumpy or bumpy in general, and if they have indentations, we can use a method called shape, and it will be using this indentation to determine where the splitting or the merging need to take place. And again, this is where these parameters can be inputted or changed in the CellProfiler software. Another important principle to keep in mind or good to know about regarding separating touching object is called suppressed local maxima. So local maxima is basically the minimum distance that's taken into consideration between two intensity peaks for separating one object from another. And so if we increase this distance, objects will get lumped together. And by decreasing this, it will just split objects. And so we can fine tune this parameter as well to make sure that objects are being identified accurately. And this is where this can be modified, again, within the Identify Primary Objects module in CellProfiler. So at this point, I would also remind everyone that it's good to fine tune, but it's also important to know where to stop. Because in an ideal world, segmentation would perform 100% well in 100% of the cases, but that does not happen. So it's important we play around with the parameters and see what's working best for our image. But the proper settings are usually achieved through trial and error. But a good stopping point would be to check if there is undersplitting or oversplitting taking place in the image. A good stopping point would be to check if this is happening in equal proportion. That is, if things are being split more or split less in equal amounts. So once the software identifies primary objects, which almost in every case is a nucleus. And why it's a nucleus? That's because nuclei usually have very robust stains such as hooks or tappy available, and they have very little staining variability, and so they can be picked up quite well. The software will produce these kind of colored images, which will show where it's identifying these nuclei, and it also will be outlining these objects with specific colors, which have different meanings. So, for example, green would be the ones which are considered valid. Yellow are the ones which are usually ignored because they are touching the edges. And if we have put in any size parameter for identifying primary objects, and if it identifies objects that lie within outside this size range, it will be highlighting those with pink. So we obviously don't want to just identify nuclei. We want to identify the whole cell and the cytoplasm as well. So the next module would be to identify secondary objects. So this is basically done by just growing out from what has been identified as a nucleus. And so we can see here the whole cells, for example, being represented in pink outlines. And it's usually for image analysis, it is seen that instead of just a membrane stain, if there's a stain included in the panel which stains the whole cytoplasm, the identify secondary object module functions better. And the principles that are used for identifying secondary object can be largely grouped into non-image-based method or image-based methods. Non-image-based method usually just identifies the nucleus and then goes on applying a fixed distance from the nucleus that spreads out from the nucleus in a donut shape. And this method is particularly useful if the panel did not have any stain for the cytoplasm or the membrane. There are some additional image-based methods as well that are used. And one of the principles that these use is a water shedding, which basically means that the software reads the image as containing some crests and some troughs, crests being the areas of the higher intensity and troughs being the one of the lower intensity. And then sort of it starts filling these troughs in a way till the point that it almost spills over to the next trough. And at this exact point of spillover would be the region where a boundary would be applied between two secondary objects. So after identifying the nucleus and the whole cell, identifying cytoplasm becomes quite easy, which would be the tertiary object. And that is just by subtracting the nuclei from the entire cell, and we get the cytoplasm area. And so by applying these three modules, identifying primary, secondary, and tertiary object, we have now performed cell segmentation. And so when we go on next to measure marker expression, we know exactly which compartment the signal is coming from. And as I already mentioned before, to check whether the segmentation is performing well or not, it's important to include modules such as overlay outline or overlay objects. This will just superimpose the segmented images on the raw images and will help us compare whether any undersplitting or oversplitting is taking place. So in the following section, I would just go over some other additional modules which are not only will be helpful to know for the pipeline that we will be building today, but also can be helpful to address some very common measurements that we tend to perform by image analysis. So one of them, for example, would be detecting speckles within nuclei. So for this, the module that's used is called an enhance or suppress feature, and this is combined with the mask images. So what the enhance or suppress feature module does is it identifies the pixels which have a higher intensity than the background, which in this case, let's say, can be fish spots within a nuclei. And the mask images will be then masking the whole nucleus. And then separating these two or subtracting these two, we can actually get a very clear identification of, for example, the fish spots. And then we can then count them or perform other additional measurements. In addition to that, CellProfiler is very, very useful for measuring many different parameters of the cell. For example, those pertaining to size or shape, such as area, perimeter, eccentricity, and so on. It also measures intensity of the objects and provides the mean, median, standard deviation of the intensity. And also, as I said, because we have performed cell segmentation, it will tell us exactly which compartment, it will inform us on the intensity measurement of each individual subcellular compartment. And not just the intensity, it also gives us information on the intensity distribution. For example, if there's an object which is brighter at the center, dimmer at the edges, or the other way around, we also get information on this from CellProfiler. CellProfiler also will tell us whether the different markers are being expressed in the same compartment or not, or in other words, whether the markers are co-localized or not. And one particular measurement, again, going back to the importance of having the spatial context or the preserving the location information that CellProfiler provides, is information on the neighbors. So CellProfiler can also measure however many neighbors that are present adjacent to our cells of interest, whether these neighbors are touching each other, the distance between the neighbors, how they are orientated, that is angle between the neighbors, and so on. So finally, after we run our pipeline in CellProfiler, we would want to save the images that it generates and also export our data. And so for that, we can click on Save Images and Export to Spreadsheet. And this will then generate a large CSV or a TSV file, depending on what we choose, which then can be used for further downstream analysis. Also, as with any other work, it's important to save our projects or pipelines. There are two different options or two different ways of doing that. We can save the entire project, and that will produce a file called .cpproj. This will contain the pipeline, the image location, metadata, and all the various different details. The advantage is everything remains saved at one place, but then this is not always portable. The other way, or a more convenient way, would be just saving the pipeline, and this will generate a file called .cppipe. This is more portable, and the great thing is when we are submitting our data or for publication, this can actually be submitted as a supplemental material. And we can determine whether we want to save both or we want to save only one of them by choosing the options in the preferences menu. So that concludes my overview, and I would imagine that there might be folks in the audience who want to learn more or who feel that, oh, this information was not enough, I need more help. So the files that I have shared for today's workshop will contain a link to the CellProfiler website, which again has a wealth of information. There are many more tutorials as well as some practice data sets available. I've also included a link to the CellProfiler GitHub for anyone who's interested in installing CellProfiler from the source. In addition to that, in the next 2 days for this I2K meeting, there will be some additional, more advanced CellProfiler related workshops as well. And again, please keep in mind that the questions that we face as researchers tend to be often not unique. And if the problem that we are facing, it's very likely someone else has faced it and has asked a question about on forum.image.sc. Please do also make use of this forum. And as you can see, that this is not just for CellProfiler, but for many other different image analysis pipelines. So I think that would be a good stopping point to see if there are already questions. And also, if there are no more questions, I think we can be heading to breakout rooms to do some hands-on exercise. Okay, so there's a question about where do I get the modules from? So let me just start up CellProfiler. So once you open up CellProfiler, this is the part where you will get option to add modules. So you just click on the plus button here and the window will show up, and you can either scan through these options, or you can just type in whatever you're trying to find, and then that will usually show up your the module will allow you to add modules. Also, please feel free to unmute and ask the questions yourself if you feel your question was not answered. Why does Run Cell Post plugin not work in CellProfiler? Can you demonstrate how the plugins work? So, okay, I see Beth has already answered it. So unfortunately, this is a very much introductory workshop. So I don't think it will be in the scope of during the scope of today's workshop to check that. But once we go into breakout rooms, we can give it a try. Otherwise, again, as I mentioned, images .sc, we can you can post your question there, or also CellProfiler conducts office hours. This might be that might also be a good resource to reach out to. And yes, CellProfiler can work on a 9612 plate. Yushan, could you specify what exactly your issue is with the Mac? Like, are you not being able to, are you getting some sort of a security message? Okay, so I think let's just head to the breakout rooms right now, because then we can address individual questions. So there are 46 participants. So should we make 4? I think.
So we have 12 breakout rooms, and you guys are gonna go in there now.
Mm-hmm. So we are almost at time. So I would just now head towards like ending the workshop with pointing out the last final couple of things. So as I mentioned, whatever that was not covered in today's workshop, or if you have additional questions and looking for more resources, the links are provided in the slides that have been shared with you in CellProfiler website. More information on how to use CellProfiler from source, other additional plugins, and more information is also available on the GitHub. And again, tomorrow and the day after, there are additional CellProfiler related workshops going on. If you're facing trouble with your images, do post your questions on forum.image.sc, and on like as a part of the CellProfiler team, we also conduct office hours. So you can also try, if you have specific questions, you can also book a session in that for us. And regarding the material that was shared today, I would also point out a couple of things. So this folder also contains a pipeline called segmentation final. So that will contain like all the parameters that you needed to input to create the final pipeline. So that's basically the answer to today's assignment. And there's also a paper for any of you if you have been a complete beginner to the image analysis. So this contains a glossary of the terms that are commonly used for image analysis. And of course, the slides for today's presentation are also included there. So that was it for today's workshop. I hope you guys enjoyed and learned a bit about CellProfiler, and will be using it more and more.
Thank you. That was really wonderful. Thank you for putting everything together so neatly.
We are very happy to do that. Okay, so have a great rest of your day, evening, or night, and hope to see you in the other CellProfiler workshops. Bye.